Very good morning, students. Welcome to GDCM SKLM YouTube channel. Myself, Pradeep. Today we are going to discussing about stem cells biology. You all know stem cells. Stem cell. What is stem cell? These are the content we are going to discussing today. That is about introduction and definition of stem cells, properties of stem cells, classification of stem cells, isolation of stem cells, clinical application of stem cells, and individual pluripotent stem cells, and conclusion. These are the contents we are going to discussing today under stem cell biology. Introduction. The stem cell, Spanish word for stem cell is cellulose madre, is a Spanish word for stem cell. Stem cells are mother cells that give rise to all other cells in the body. That means these are the pioneer cells which can differentiate into any other types of cells in the body. If you take an adult body contains nearly 200 different types of cells. All these 200 different types of cells coming from one cell, that is zygote, which is called stem cell. So stem cell is the pioneer cell which can generate all other types of cells present in the adult organism's body. They are cells found in all multicellular organisms that can undergo mitosis to give rise to specialized cells or more stem cells. That means every stem cell can produce specialized cells or differentiated cells along with same type of cells. For example, if any stem cell divide through mitosis, it produces two cells. Among the in these two cells, one cell behaves like a stem cell and another cell divides, differentiate and form into another type of cell. So in every division, the stem cell divides, produce two cells, one cell retain its property and another cell become differentiated and produce another type of cell like liver cell or kidney cell or brain cell or nerve cell, any type of cells. They are considered as a blank microchips because they are considered blank microchips. Why they are considered as blank microchips? Means all the stem cells have not specialized. That means they don't possess any specific function. So that's why they are called blank microchips or like blank like white paper. That means they doesn't possess any specific function. So their stem cells are considered as blank microchips that can be programmed to perform particular task. We can later, after differentiation, the stem cells gain specific function. They serve as a repair machine for the body and also they repair the body also. For example, if there any wound is formed in the body or any tissue is damaged, that tissue again regenerated due to the presence of stem cells. So stem cells are blank cells. They can have the potential to differentiate any other type or cells or tissue present in the body and also they are involved in the repair mechanism also. Definition. Stem cells are defined as the cells that have clonogenic and self-renewing capability. The clonogenic, what is clonogenic? Clone means what? These are the group of similar type of cells that are produced from single cell. So single cell upon multiple division produce group of similar type of cells. Those are called clones. That's why the stem cells are called clonogenic and self-renewal capacity. Self-renewal means the same type of cells can be produced again and again. For example, if any cell is damaged, then that type of cells is reproduced by this type of stem cell. That is self-renewal. It can reproduce itself capability and differentiate into multiple cell lineages. If you take this stem cell divide through mitosis and produce same type of cell, that is stem cell, and another type of cell called specialized cells. That means specific function differentiated cells. So in each and every division, the stem cell produces two cells. One cell retains its property like stem cell, that is self-renewal capacity, and another cell differentiates into specific function through differentiation process. In this way, every stem cell in our body have self-renewal and also produce different type of cells. So a cell that has the ability to continuously divide and differentiate into various other kinds of cells is also known as stem cell. This is the definition for stem cell. Next, properties of stem cells. These are the different properties of stem cells. Stem cells are unspecialized cells are called blank cells. That means, as I already told, stem cells doesn't possess any specific function. So they have the potential to regenerate any other type of cells present in the body. So they are called blank cells. Why they are called blank cells? Because they doesn't possess any specific function. Don't possess any function, specific function. That's why they are called blank cells. Asymmetric cell division. Why asymmetric? This is asymmetric cell division. Every stem cell divide asymmetrically. Stem cell divide in what manner? Mitosis. Mitosis division occurs. It produces two cells. That produce two cells. The daughter cells, both the daughter cells have the don't have the same properties. That means that's why it is called asymmetric division. Asymmetric division means the produced two daughter cells, one cell retain its property and another cell become differentiated. So the produced two cells have different properties. One is stem cell property, another cell possess differentiated into another type of cell and get specific function. So it is asymmetric cell division. This is the meaning of asymmetric. Asymmetric means the produced daughter cells both don't possess similar properties. That means one cell retain stem cell properties and another cell become differentiated. So the produced two daughter cells possess different function. That's why it is asymmetric cell division. And stem cells can divide and renew themselves for long period of time. Why it is renewal? Because after division, two cells produce, one of the cell retain the stem cell property. So this is known as self-renewal capacity. So each and every stem cell possesses self-renewal capacity, but differentiated cell doesn't possess self-renewal capacity. So again, stem cell need to come and renew the other type of cells, but stem cells can renew themselves itself, and stem cell can divide and become specialized cell types of the body. That means stem cell have two property. It can renew itself and also produce another type of differentiated cells also. Stem cell can replace dying or old damaged cells. So the stem cells also replace the old or damaged cells in the body. And relocation and differentiation are abilities of stem cells to migrate to where they are needed in the body and specialize into particular type of cells. For example, one stem cell is present in the liver and damage occur in the kidney. Then the stem cell migrate to that kidney part and specialized into kidney cells and produce kidney. That means the stem cells can migrate any other part of the body and they regenerate whatever the cell required in the body. That means they have the capacity to may migrate from one place to another place and also replace the cells whatever the damage in the body. These are the several properties, different properties of the stem cells. Cell division in stem cell. It is a stem cell, a single cell that can replicate itself and also produce different types of cells. As I already told, the stem cells have the asymmetric cell division. That means after cell division, it produces two different types of cells. One is self-renewal capacity. That means similar type of cell. That means stem cells. Another type of cells are differentiated. So here stem cells after division produce two different types of cells. So it is asymmetric division. One is same property possess. So one cell is possess similar properties as the stem cell, and another is differentiated cell. These are the cell division potential of the stem cell. Stem cell classification. There are different types of stem cells based on their potency. Potency means some stem cells can give entire organism, whereas some stem cells give only particular type of germ layers, either ectoderm or endoderm or mesoderm. Whereas some stem cells can generate only specific lineage or specific type of germ layer cells. And like this, based on their potency, the stem cells are categorized into different types. Those are embryonic stem cells, fetal stem cells, and adult stem cells. These are the different types of stem cells. Those are embryonic stem cells, fetal stem cells, and adult stem cells. Embryonic stem cells. Embryonic stem cells are two types. Those are pluripotent and pluripotent. Totipotence means total potential. That means each and every totipotent cell can generate entire organism. For example, zygote. Zygote is the totipotent stem cell. It can generate entire organism. So it is a totipotent stem cell. Another one is pluripotent stem cell. Pluripotent stem cell has the potential. It cannot produce entire organism, but it can produce cells of the three germ layers. It can produce ectodermal cells or endodermal derived cells or mesodermal derived cells, but cannot produce placenta. Due to the lack of placenta producing capacity in the pluripotent cells, they cannot generate entire organism. But totipotent cells can produce entire organism. Pluripotent cells can produce only three germ layer cells present in the three germ layer cells. That means they can produce any type of cells that are produced by the three ectoderm or mesoderm or endoderm, but cannot produce, pluripotent cells cannot produce placenta. If placenta cannot produce, it cannot produce whole organism, because in animal cells placenta plays a vital role in the development of embryo in the mother womb. So these pluripotent cells have no capacity to generate placenta. So pluripotent cells cannot generate entire organism. So pluripotents have the certain limitation to produce entire organism. And multipotent cells, these multipotent cells can produce only single germ layer, either ectodermal germ layer cells or endodermal germ layer cells or mesodermal germ layer cells. Only that means their potency is decreasing from totipotent to pluripotent and pluripotent to multipotent. Pluripotent cells can produce any of the germ layer cells, either ectoderm or mesoderm or derm, but multipotent stem cells can produce only single germ layer producing cells. For example, ectodermal cells. From ectoderm, there are several organs formed. For example, nervous system, skin, and all these cells can produce it from ectoderm. So only that ectodermal derived cells only develop from multipotent stem cells. And oligopotent stem cells. These oligopotent stem cells can produce only specific lineage specific cells. That means, for example, if you take blood cells, all blood cells derived from hematopoietic stem cells. So if you take blood contains several types of cells: B cells, T cells, erythrocytes, and platelets, and all these different types of cells produced from hematopoietic stem cells. These hematopoietic stem cell is the specific best example for oligopotent stem cells. So they can produce only specific lineage specific cells. And unipotent stem cells. Unipotent stem cells can produce single type of cell. For example, liver cell. Liver contains liver stem cells. Whenever liver cells damage, that damaged cells is replaced by that unipotent liver stem cell. So these are the different types of stem cells based on their potency. Totipotency means total potential. That means these cells can produce entire organism. Pluripotent stem cell is the stem cell which can generate any cell of the three germ layers. Multipotent stem cell is the stem cell which can generate any particular type of cell of a particular germ layer. Only single germ layer cells can be produced. Oligopotent lineage specific cells, and unipotent stem cells, these are tissue specific stem cells. So if you take liver cell, liver contains each and every in our adult organism, each and every tissue contain tissue specific stem cells. All these tissue specific stem cells are called unipotent stem cells. In this way, during development of embryo, the potency of the stem cells decreases gradually from totipotent to unipotent. These are the classification of different types of stem cells. Classification of agility to totipotent. Each cell can develop into new individual cells from early 1 to 3 days of embryo. That means after fertilization, up to 3 days, the embryo have the capacity to generate, have the potential to generate entire organism. Then pluripotent stem cells. Pluripotent stem cells can form only over 200 cell types. If you take adult body contains 200 different types of cells. That means all these 200 different types of cells derived from three germ layers, either ectoderm, mesoderm, and endoderm. So any germ layer cells can be produced from pluripotent cells except placenta. Some cells are the last 5 to 14 days. If you take embryo, 5 to 14 days embryo can give this pluripotent cell. Multipotent, cell differentiated but can form a number of other tissue. That means only single germ layers can be produced. That means fetal tissue, cord blood cells, and adult cells. Unipotent, able to contribute to only single mature type of cells like muscle stem cell or blood cells or this type of unipotent stem cells. Classification of stem cell, this is example. If you take fertilized egg and up to 3 days, this is totipotent. Up to 15 days, pluripotent. After that, multipotent, then unipotent, then lineage like that, oligopotent and unipotent like milk. Next, classification on the basis of the source, embryonic stem cell. If you take stem cells from embryo, those are called embryonic stem cells, and embryonic stem cells are mostly totipotent or pluripotent. Whereas adult stem cells are somatic stem cells. Adult stem cells present in specific tissue. For example, you take liver contains liver stem cell, kidney stem cell, heart stem cell. Though they are contain adult stem cells. So all adult stem cells are oligopotent or unipotent, whereas embryonic stem cells are totipotent or pluripotent, whereas adult stem cells or somatic stem cells are oligopotent or unipotent. Embryonic stem cells found only less than 2 weeks. That means embryo. These can found only embryo before 2 weeks. 2 weeks is embryo in the development of an embryo. Embryonic stem cells are the most versatile because they can become any cells in the body, including fetal stem cells and adult stem cells. That means embryonic stem cells have the greatest potential to can generate any other type of cells present in the body. Embryonic stem cells are taken from inside the blastocyst. Embryo contains blastocyst and blastocyst contains cells. These embryonic stem cells, from blastocyst we can derive these embryonic stem cells, early stage of embryo. The blastocyst is a ball of 50 to 100 cells and is not yet implemented in the womb. It is made up of an outer layer of the cells, a fluid filled space, and a group of cells called the inner cell mass. Embryonic stem cells are found in the inner cell mass. These embryonic stem cells are found in the blastocyst inner cell mass. This is the fertilized egg, and this is totipotent cells. And after further division form ball-like structure called blastocyst. Blastocyst contain inner cell mass. These inner cell mass contain collecting these inner cell mass, and they can culture and produce any other type of cells present in the adult organism. Adult stem cells are somatic stem cells. Adult stem cells are found in the human body and in umbilical cord blood. They can found in the human body. Each and every type of tissue present in the human adult organism contain these adult stem cells or umbilical blood cells, also known as the tissue specific stem cells. They are also known as tissue specific stem cell. That means they can generate only particular type of tissue only. The most well-known source of adult stem cells in the body is the bone marrow, but they are also found in many organs and tissues, even in the blood also. But isolation of these adult stems is very difficult because they are present in very minute quantity. So isolation of these adult stem cells is very critical, very difficult. Adult stem cells are more specialized since they are assigned to a specific cell family such as blood cell or nerve cell. That means they can generate only particular type of cell only because they are highly specialized. Recently, it was discovered that an adult stem cells from one tissue may act as a stem cell for another tissue. This is sometimes recently it has been identified that some adult stem cells, specific adult stem cells, can also produce another type of cell. For example, blood stem cells can also produce neural cells based on their programming. Location of somatic stem cells in the body: blood vessels, brain, skeletal muscles, bone marrow, liver, heart, and that means each and every tissue contains their specific somatic stem cells or adult stem cells to regenerate whenever the part is damaged. Embryonic stem cells. These are embryonic stem cells, but embryonic stem cell, adult stem cell has a limited capacity to regenerate. That means they can produce only specific type of cells, but embryonic stem cells can produce any other type of cells present in the body. That means if you take embryonic stem cell and you want to culture, we can produce more than 200 different types of cells present in the body. If you take embryonic stem cell, you can produce liver cell, you can produce skin cell, you can produce blood cells, and also you can produce neuron cells also. So embryonic stem cell have the greatest potential or greatest capacity to regenerate into any other types of cells present in the body. So these embryonic stem cells have the great applications in the regenerative medicine. Adult stem cells, what they can do? blood stem cells differentiate and produce different types of blood cells. Bone cells can produce only different types of bone cells. That means adult stem cells can give only specific type of stem cells only. Differentiated cell, but embryonic stem cells can give any other type of all types of cells present in the body. Isolation of human embryonic stem cells. If you take, how can we isolate embryonic stem cells? This is egg sperm fertilization. In vitro fertilization gives a zygote subjected to cell division and produce blastocyst. It contains inner cell mass. These inner cell mass isolated for using micro injection process method, and these isolated inner cell mass cultured into media. After culturing the media, differentiated into any other type of cells present in the body. In this way, embryonic stem cells have great potential to can produce any other adult cells present in the body. This is the process of isolation and regeneration of embryonic stem cells into differentiated cells. Clinical applications. What are the clinical applications? Possible treatments by the stem cell. Embryonic stem cells can be used for the baldness. That means baldness can be treated using these stem cells. And Alzheimer's disease and sclerosis, multiple sites of cancers. Cancer can also treat with using stem cells. And spinal cord injuries, bone marrow transplantation, blindness, Parkinson's, so many diseases we can cure using these stem cells. So stem cell biology or stem cell therapy has the greatest applications in the human therapeutics or human medicine. Next, individual pluripotent stem cells. What are these individual pluripotent stem cells? These individual pluripotent stem cells are the stem cells that are produced from adult stem cells. Generally, there are so many ethical issues isolating the embryonic stem cells from embryos. So, in order to overcome that, we can generate embryonic stem cell-like cells from adult cells using individual pluripotent stem cell method. Individual pluripotent stem cells. The cells from the body. This is adult organism. We can take adult cell. That means differentiated cells from the body. That means adult cell. That means they have no pluripotent, no stem cell property. If we isolate these cells, genetic reprogramming, add certain genes to the cell. That means whatever the cells that are responsible for the maintenance of stem cell properties, those genes isolated, the genes introduced into this adult cell through DNA technology. Then they become individual pluripotent stem cells. So, culture of individual pluripotent in the lab, differentiate they can produce. So, individual pluripotent cells can be produced from adult cells by injecting or transferring genes that are responsible for maintaining the stem cell properties. To carefully study the embryonic stem cells, property is governed by particular type of genes. We can find that genes and isolate that genes and introducing those genes into adult cells, and we can produce and can generate stem cells from adult cells. So these are called individual pluripotent stem cells. Individual pluripotent cells also have the property to differentiate into another type of cells present in the body. Scientists turn human skin cells into stem cells. This is the human skin cell in the adult cell. These adult skin cell converted into stem cell by transferring these genes. So by inserting just 4 genes, that is OCT4, SOX2, KLF4 and MYC. These 4 genes are responsible for the maintaining of stem cell properties in the stem cell. So isolating these 4 genes and inserted into vector, cloning vector, and transfer these inserted recombinant vectors into skin cell, and the skin cell converted into pluripotent stem cell, and these pluripotent stem cells can be used for the generation of any other type of cells present in the body. This is one type of individual pluripotent stem cells. Nobel Prize in the Medicine. The Nobel Prize in Physiology or Medicine 2012 was awarded jointly to Richard B. Gordon and Seini Yamanaka for the discovery of the mature cells can be reprogrammed to become pluripotent stem cells. So, individual pluripotent stem cell technology developed by these 2 scientists, and they received Nobel Prize in 2012 in the medicine. Conclusion. So, stem cell research is complicated and rapidly changing. Today's medicine generally tries to support or treat injured tissue and organs, but stem cells may someday simply replace them. Nowadays practice is to just treat or injury tissue. just generally tries to support or treat injured tissue. Whatever the tissue is injured, that tissue again use drugs used for the treatment of that tissue. But if you use stem cell therapy, they completely replace the damaged tissue. There is no need of separate drugs for curing the injured tissue. So stem cell therapy has the greatest potential, and stem cell therapy is considered as like a soldier with a weapon. Only if the soldier weapon technology and bullets stem cells cell are in our hand, then fight will turn into our favor. So in this way, stem cell technology offers a greatest chances to future therapeutics or future treatment. It's the greatest weapon for the treatment of disease. So this is about all about stem cells and its biology. Thanks for listening.